Students, there are two types of DNA. We can simply call them as prokaryotic DNA and the eukaryotic DNA. Otherwise, you can call them as circular DNA and the linear DNA. So some of the differences I have written here. Let's go through them. The prokaryotic DNA is found in the cytoplasm as well as mitochondria and plastid, because we need to remember in the 11th class also we have done that the structural composition of mitochondria, chloroplast as well as the prokaryotic cells are quite similar, and the easiest way to understand this is the presence of the 70S type of ribosome. So when we look at the prokaryotes, they have a circular DNA inside, and when we look at the mitochondria and chloroplast also, they also carry the circular DNA inside. So all the prokaryotic DNAs or mitochondrial and plastid DNAs are going to be circular in nature. So they can even be called as prokaryotic type of DNA. But when we talk about the eukaryotes will always have linear type of DNA, and it is present inside the nucleus. The prokaryotic DNA obviously is going to be less in amount because it's a small organism, so small would be the genome. We have already covered the length also, if you remember. If you pay attention to the length of the DNA of Escherichia coli, it's a prokaryotic organism. It has 4.6 10⁻⁶ base pairs. And when we talk about the diploid amount, it would be 6.6 10⁹ base pairs. So just see the difference between the powers here, 10⁶ and 10⁹. The reason is that the E. coli is a prokaryotic organism and human being is eukaryote. So the amount of DNA even in the mitochondria and plastid would be quite small. So because these DNA are going to be less in amount, but this is going to be bulkier, it is more in amount and is inside the nucleus of the eukaryote. The next difference we can remember, it's circular, this one is linear. Remember, this is going to be double stranded and this also going to be double stranded, because we are talking about prokaryote, not the virus, because only virus can have single stranded DNA or single stranded RNA. The next difference we can understand is that the DNA found in the prokaryotes is devoid of histone proteins. So what are histone proteins? Basically these proteins help in packaging of DNA. So if histone is absent, the DNA would be called as the naked DNA. So the naked genetic material is found in the case of prokaryote. But when we talk about the eukaryotic system, histone proteins are present which help in the packaging of DNA, so it will not be called as a naked type of DNA. Then remember that the prokaryotic DNA codes for very few proteins. Reason, because if genome is small, if the genetic material is small, it will code for very less protein. And in the case of eukaryote, because the length of the DNA is more, so the more the length of DNA, more proteins it will code for. Then introns are absent, but the reality is introns are present in very less amount in the case of prokaryotic DNA, and they are present in abundance, and that's why the eukaryotic genes are called as split genes. If you remember, I have talked about the introns and exons. If you that same concept in the case of the H and RNA, please go through those notes. You will find this concept was there. Otherwise, in detail, we are going to do it later in this chapter only. Then the next difference we can understand is, remember a few minutes back we talked about the ratio of G plus C and A plus T. So same thing now. The ratio of A and C is going to be more compared to, G plus C is going to be more compared to A plus T in the case of the prokaryotic organisms, and in the case of eukaryotes, the amount of A and T is more compared to the amount of G plus C. Remember, G plus C provides more stability because there are 3 hydrogen bonds. A plus T provides less stability because there are 2 hydrogen bonds. So because primitive organisms lived in a harsh environment, or harshest of all the environments, so we can link in that manner that G plus C is more in the prokaryotic organism, and A plus T is more in the case of eukaryote, because eukaryotic system has developed more things for stability. For example, histone proteins are there. Then the next is repeated sequences are absent in the case of the prokaryotic organisms, DNA, and the repeated sequences are very much present. We'll be learning in this chapter in the case of humans also. Eukaryotes DNA carry a large amount of repeated sequence, and remember, very less proportion of the DNA is functionless in the case of prokaryote, but in the case of the eukaryotes, a large proportion of DNA is functionless, and the DNA is called as junk DNA. So you can learn some of these differences. The next thing to start with is the structure of the polynucleotide DNA or RNA. So how does the structure look like? Let's quickly go through this because I want you to understand the various bonds which are involved here. I have seen you making mistakes in the assignments regarding these bonds even in the last class. Nucleotides are the basic unit of nucleic acid. So nucleotides have three components: nitrogen base, pentose sugar, and phosphate group, ribose sugar in RNA, deoxyribose sugar in DNA. There are two types of nitrogen base: purines, two further types; pyrimidines, three types. Cytosine, adenine, guanine are present in both DNA and RNA, but thymine is present in DNA, and uracil is present in the case of the RNA. Nitrogenous bases is linked to the pentose sugar by N-glycosidic bond to form a nucleoside. Depending upon the type of the pentose sugar, and the nucleosides are of various types. They can be ribonucleosides and deoxyribonucleosides. Uracil produce nucleoside with only ribose sugar, and thymine forms only with the deoxyribose sugar. The different nucleosides are, so you can learn these names: adenosine, deoxyadenosine, this is found in RNA, this is found in the DNA; guanosine, deoxyguanosine; uridine, deoxythymidine, difference between DNA and RNA; cytidine and deoxycytidine.